All right, students, welcome back. This is the last part of chapter 9. In the previous two sections of this week, we covered concentrations of solutions and dilutions of solutions. Those two sections have a lot of calculations. In this last part, properties of solutions, you will have a little reprieve. There will be more concepts here than calculations. In this part, we will learn to identify if a mixture is a solution, a colloid, or a suspension. A solution is something that we've been working with throughout this semester already without mentioning it. Everything that we've seen is basically a solution. Like for example, pictured here, this sodium chloride saline solution is a solution mixture. So in this section, we will learn two new types of mixtures called a colloid or a suspension. And then we'll learn how these particles will affect something called the osmotic pressure, a natural occurring phenomenon, and how that is related in the human body in a concept called dialysis. And so let's first start to describe these three types of mixtures. As our prime example in the solution here, like we mentioned earlier, is a saline solution, but the solutes are sodium and chloride ions. Our prime example of a colloid solution here would be a glass of milk. And our prime example of a suspension mixture is a glass full of mud. Okay, so take a look at these three types of mixtures, and can you see what their differences and similarities are? Let's first think about transparency. Which of these is transparent and which is definitely not transparent? So yes, a solution particle, light can go through it, you can see through it, right? So we call that transparent. Whereas a colloid solution is not transparent. Sometimes it could be, but mostly not transparent. And lastly, of a suspension, can you see through the glass of muddy water? You cannot, right? So we call that what? Opaque. So now let's go back to a different concept. Remember we learned in the beginning of the semester, we can describe a mixture as homogeneous versus heterogeneous. What do you think? Which one of these are homogeneous? Which one is heterogeneous? Remember, homogeneous is if we take a sample of that solution from one part versus another part, they are equally the same amount of things in them. So if you said that the solution and the colloid are homogeneous, you would be correct, right? If you took a straw and you sipped on the saline solution at different locations, if you took a straw and you took a sip from the milk in various locations, it would not matter, right? So it would be homogeneous throughout. Whereas a suspension, it's heterogeneous. Another way to think about that, remember in a homogeneous solution, you cannot tell the solute from the solvent. For example, in the NaCl solution, you can't tell the NaCl from the water, right? Or in the milk, you can't tell the protein particles in there from the water. Whereas in the suspension, you can distinctly see the chunks of dirt from the water. So that's why it's heterogeneous. And most importantly, a key difference between these types of mixtures is in the way they can be separated. Remind ourselves that a solution particle could be something like an ion or a small molecule, like sugar molecule. Whereas a colloid particle could be something much bigger, like a protein, a particle with lots and lots and lots of atoms in it, so it's much bigger. Whereas a suspension is the largest particle, and so large that you can actually see with your eyes. These two you cannot see with our eyes. This one you may be able to see with your eyes. Okay. Sometimes colloids that are stuck together can become a suspension so that we can see it with our eyes. So basically, the difference here is the size, right? So if you had a mixture of all three, solution particle, colloid particle, and suspension particle, so if you were to pour it through a filter, let's say a simple paper filter, in this paper filter, there are holes that allows a certain size of particles to flow through. Imagine this filter paper like a screen door. Like air can go through, but like bugs cannot go through, right? So that's why we have that screen door. Or another example is like wearing a mask. When you wear that mask, you are preventing aerosol droplets from other people's coughing from coming through and infecting you, right? But you are allowing smaller air particles to go through so that you can breathe. So a filter is basically that. Different filters will have different sizes of holes to allow different sizes of particles to flow through. But in general, a paper filter will allow what to flow through, as you can see from this image here. It will allow the solution particle and the colloid particles because those are small enough to flow through. What is being kept behind is the suspension particle, okay? Where is a filter paper that you see in everyday life? Like if those of you who drink, let's say, coffee, you can see this. When we filter coffee, the coffee grinds are stuck on top of the filter paper, but all of the caffeine and even the color brown and the water will flow through, right? And those we will consider solution particles, and coffee grinds on top we will consider suspension particles. Now, so then you may ask yourselves, is there a way to separate these two types of small particles, the solution particle and the colloid particle? And indeed there is. There's something called a semi-permeable membrane. Okay, let's break up this word. Semi meaning half, kind of, right? Semi, like half and half. Permeable means going through, being able to permeate. So something, a membrane, a filter, if you may, that allows some things to go through. And every semi-permeable membrane will have its own limitations. But in general, let's say this membrane will only allow solution particles to go through. So notice if you put this solution, if you put a mixture in this bag and you put it into a solution of water, over time, only solution particles may escape, but the colloid particles and the suspension particles will stay inside. Okay, so that's an example of a semi-permeable membrane. Only solution particles can go through because they are very small. And this image over here also highlights one more similarity and difference between these three particles. Just like a solution of muddy water, right? If you let it sit over time, what would you see? You will start to see that dirt will settle at the bottom and then the water is basically clear on top. So a suspension particle will settle down. So you can see this sometimes in medicine. When you read a label, it says to shake well before use, right? Like an antacid mixture, you need to shake well to make the settled particle become a suspension again. Or else, if you take that medicine, you might just be taking the water and not the drug itself because it has settled down. Whereas if a drug is a solution or a colloid, you do not need to shake it well because it is always going to be dissolved in the water and always suspended throughout the solution. All right, so with that, let's go back and fill in our Venn diagram. All right, so in terms of filtration, we said that suspension particle cannot go through filter paper or a semi-permeable membrane. I'm going to abbreviate that as SM. What about the colloids? And lastly, the solution particle can go through semi-permeable membrane. And since we're here, let's also note that this does not settle, and heterogeneous means it will settle down over time. Now let's talk about a natural process called osmosis and its related osmotic pressure. And so this is a process that is found in plant and animal cell walls that is super important for biological processes, and it boils down through the movement of water. So in osmosis, the principle is that water will move from a solution that has low concentration of solutes to a high concentration of solutes. And the water is basically trying to flow through this side because it wants the two sides to be equal. So if there's no solutes in this side and lots of solutes in this side, so the solutes concentration is very high over this side, right? So and how do we make these sides equal? Is we will dilute this side, right? If we increase the volume of water, the volume of the liquid in this side, if you increase the volume, then without increasing the solute, the concentration of the solute will go down. So that's the driving force of why water is moving towards this side, to make the two sides equal to each other. And another way to make the two sides equal to each other is for the solute to, on this side, the concentrated side, to move over to this side. That will also work, but in this case, the semi-permeable membrane is so small that it only allows water to move. So because of that, only osmosis is happening here. So water molecule will keep on transporting to the more concentrated side. So over time, there's more water on the one side, increasing the height on the sucrose solution. But eventually, the height of the sucrose solution will create a sufficient pressure to equalize the flow of the water between the two components. Okay, so at that time, the force that's pushing water over here versus the force that's pushing down on the solution will be equal so that no more water will come through. This is called the osmotic pressure. And the osmotic pressure depends on the concentration of the solute particles in your solution. The more particles dissolved, the higher your osmotic pressure will be. The more salt you have in one side is the more water you want to move over. And that amount of water moving over is essentially related to the osmotic pressure. Okay, now let's apply that concept in our biology. Because remember, our cell membranes are basically biological systems that are semi-permeable membranes. Osmosis, therefore, is an ongoing natural process that's always occurring. For example, here we have a blood cell. If we put this blood cell in a concentration of 0.9% mass over volume of sodium chloride, it will have the same osmotic pressure as our blood cell. Alternatively, if we use 5% glucose, again, mass over volume percentage, we will also have a solution that has the same osmotic pressure. And so we call this type of solution isotonic solution. And in isotonic solution, again, the osmotic pressure outside, which is the solution, is equal to inside, which is your cell. Okay, because of this, there is no net movement of water, and then therefore we will have a normal cell. Now let's see what happens when you put that same cell into a solution that has less solute. Can you imagine what will happen? If it has less solute, where will the water be flowing? Okay, again, inside is higher concentration than the solution outside. So where is water moving? Water is moving from outside into, right? So the inward movement is bigger than the outward movement. Very good. This is called a hypotonic solution. Hypo meaning less, right? So that solution has less solutes. In a hypotonic solution, because you have less concentration outside and then the more concentrated inside, water flows into the cell. Now, what happens, do you think, if the water flows into the cell too much? The cells will swell and eventually burst. So that's a bad thing, right? Now, on the other hand, let's imagine if you put your cell in a solution where there's higher concentration of solutes, so higher osmotic pressure. What will happen? The water will flow from low to high concentration, right? So more water will flow out than flowing in. In that case, you're kind of depriving yourselves of water, right? So the cell will then shrink. So that's called hypertonic solution. Hyper meaning more. The solution has more solutes, higher concentration. Okay, so both of these scenarios are not good. In hypotonic solution, your cells will burst, and that term is called hemolysis. And then in this hypertonic solution, the cell will shrink, called crenation. Both of that is not good. So that's why when you go to the hospital and you need an IV, that IV is osmotically balanced so that your cell wall will not be harmed. And our last concept here is dialysis, and dialysis is very similar to osmosis. So in dialysis, a semi-permeable membrane will allow solid particles and ions, as well as solvent water molecules, to pass through and retain any big particles like a colloid or a suspension particle. As you can see here, over time, these smaller particles go out. This dialysis bag will keep the rest. So in this particular bag, the colloid particles that are allowed are protein and starch particles. And one detail I want to point out to you here is that most students think that the solution particle will all flow out, right? And then there will be none inside. That's not actually true. It will flow out until the concentrations of the solution particle outside is equal to the solution particle on the inside. So that means the solution particle will exist in both locations. And the colored particles will just exist on the inside, none on the outside. And so with that, let's do a concept check. An aqueous mixture containing starch, so we said is a colored particle, and glucose, which is a solution particle, is placed in a dialysis bag and immersed in distilled water. Which of the following correctly describes the location of the indicated substance after some time of dialysis? Let's go over the options here. A is will you only see starch inside? Okay, that's true, right? Starch will only be on the inside. Okay, so that might be an answer. Let's keep on going. Do you see that starch is inside and outside? That's not true because starch is a colloid, and so in a dialysis bag, it cannot escape. Okay, so no outside. So B is not true. So C, it says glucose inside only. So that's a common misconception that I just mentioned earlier, right? Glucose will go outside until it is equal in concentration inside and outside, meaning there will be some on the inside and some on the outside. So the word only here is false, which is why C and D are both wrong. So the answer here is A. All right, so I want to end our discussion here applying dialysis in the health field. As you may know, the fluids of our body will undergo dialysis by the membranes of the kidney. Our body goes through a lot of processes where it will create waste products, and these waste products needs to be expelled somehow, right? And so they are in our blood first. So our blood will go through the kidneys, and in that kidney there are different cells that will dialyze out these particles, these waste particles. So these waste particles like amino acids, glucose, urea, etc., will move through these membranes outside of your blood where it's highly concentrated, move into these other compartments that has less concentration of those things. So that's why it's called dialysis. And eventually move along, and we will remove it from our body by peeing them out. Okay? Now, if your kidneys aren't working, then you would have to undergo what is known as hemodialysis, where you are connected up to this machine that does the job of your kidneys. So this machine will pump out blood. Again, in your blood, you have these waste products that your body needs to get rid of, and then that blood will go through this coil, which is submerged in a dialysate, or basically a solution that has low concentrations of your waste. So therefore, dialysis happens where the waste solute particles will move out of the semi-permeable membrane and into your into this solution. Now, as it goes through this coil, over time, more and more of these waste products being transferred out. And by the end of that coil, your blood is basically clean of these waste particles. And then, so therefore, it pumps it back into the body of the dialysis patient.